so let's start with the material. We'll just stop at one slide, and then we will do these iClicker questions. There will be a lot of them today because of this material. So just make sure that the key part for the part one is that know each type of these membrane, like the proteins, how they're inserted into the membrane. The soluble is just only one. It just goes in there with the N-terminal signal. Type 1 is similar to that. So look at the supplementary video, follow the Lightboard videos, and just make sure that you can draw, if a linear sequence of topology is given, you can draw it on the membrane of the ER. Also, very interesting, that just make sure that if suppose one of these membrane protein actually is transported onto the plasma membrane that will learn, then which part should be the outside the cell and which side will be inside the cell. Know that. Remember that all the cytosolic part, even if this becomes a membrane protein, is going to be on the inside. The lumen is equivalent to the exoplasmic phase, the white space in between the organelle and the plasma reticulum is Golgi. They are all single membrane. So the inside lumen, the white space that we saw in the previous classes, previous chapters, previous unit, the lumen is going to be the exoplasmic phase, whereas the cytosolic is the towards the cytosolic or the cytoplasmic phase. So another thing is that I am still not discussing anything about the exam 1 because I still have 2 illness-related makeups to give. So it's open. You can see that I have to make 2 different exams for that, and so I will discuss that once those are done. But you can always office hours, and that if you just want the study strategy, make sure that you pick up that Excel file, the module part study technique, and then send it to me. Then we can just figure it out. There could be some question that did you watch the Zoom review also for my online class? So just ignore those. There was the exam review was in class. It was recorded, available through Zoom, but there was no separate Zoom. So just to make sure that you know that. So that's pretty much it. But we will discuss about that once we are done. I know that it is a little bit frustrating, but you can always ask for office hours though. All right, so this is basically the idea that make sure that you know this. You put this one on a membrane, and if a membrane protein is given on the other hand, so what do you mean by that? Give you a protein like this, you should be able to write that in a linear topology. That's I mean, so back and forth and do that. And this is the last slide of the part 1 that we did not go through. That this is the hydrophobicity plot. So what we mean by this is we have a linear scale, and the scale starts at 0, and above that you have a scale of 0, 1, 2, 3, 4, and below that you also have the same one. So what it considers that if you have a protein, it essentially considers about 20 stretch of amino acid, and then it calculates that specific numbers are not required, and then it calculates that the net hydrophobicity of that, whether there are more hydrophobic residues in those amino acids or they are hydrophilic. Hydrophilics are usually charged and polar amino acid, whereas the hydrophobic should have the big rings like phenylalanine, tryptophan, histidine, those kind of things. And those are the hydrophobic ones. And then based on that, this graph is generated. So essentially what happens that if you can see that if you remember that there was lots of hydrophobic sequences followed by some hydrophilic sequences, charged sequences in the signal sequences. And of course you can see that there is a peak above that 0 line. So anything above that is a net hydrophobic region of the protein. So remember that. So then you have the stop transfer signal. This is a membrane protein type 1 because it has a signal sequence. So here, this interacts with the translocon, and that's also hydrophobic. So you can see there is a prominent peak in there. This is a hydrophobic peak. Now, there are some tiny peaks in there. We usually do not consider those because we just consider the biggest ones. Here you have just only one signal anchor sequence because see that protein starts from here, but there is no signal sequence and it's a signal anchor sequence. And here you have a transmembrane sequence, and each one of them is a transmembrane path, and you know that it has to interact with the hydrophobic part of the fatty acid tails, essentially. That's what it is. So usually the type of question that is given, we will give you a plot, and from that you will be able to tell that whether that protein, how many membrane spanning domains are there. So all you have to do that count the number of peaks above the zero line. The number of peaks above the zero line is equivalent to that you have those hydrophobic domains which will pass through the membrane. If you have essentially a domain which is at the very beginning of that, say that right here at the very beginning, remember that that should be the signal sequence. Here I cannot tell that it's a signal sequence or not because this is close, but it really cannot tell. But if it starts really at the very beginning, I will be able to tell that this is a signal sequence. Here it is definitely not, and here I guess that is not as well. So these are the things. But that is, so the question is could be that do I have to count these tiny ones? And how do I know that I do not have to count this one and I'll count this one? So for your exam purpose, I'm not going to trick you on that. So anything above that line is hydrophobic. There I will not put like these tiny things in there so that it confuses, or you can think that, okay, here you have 1, then you have 2, 3, 4, 5, 6, 7. They are above that. So you should not have that confusion. So just straightforward above the line, hydrophobic domains passes through the membrane or could be the signal sequence if it is all the way at the very beginning, or yeah, that's it pretty much. And the ones which are below, those are usually the pixoplasmic luminous or cytosolic hydrophilic residues, which are like loop-like structure in a multi-domain, like a multi-pass transmembrane protein. And this is essentially what you're seeing, that this is the signal anchor sequence, and the rest of the part is going to just flop around essentially in the lumen or in the cytoplasm, depending upon the type of the protein to treat. So know this very well. All right, any questions from that? So let's do some quizzes. So we have already did that. Just remember that in absence of any signal targeting sequence, the expected location for the protein is always cytosol, or for any other endoplasmic reticulum, lysosome, Golgi, secretory vesicles, you need a signal sequence or targeting sequence. So as for your nucleus, mitochondria, chloroplast, peroxisome, all of these will need a sequence of different type. Only the ones will not have anything is the cytosol. All right, so let's go to the next one. All right, so this is in between lines. So if you have studied that, should be able to answer. Two-minute each question, because we have covered these material a day before. So for each question, you have 2 minutes exactly. more seconds. All right, stopping it. Let's see. All right, so you can see that the C is the correct answer, but it's just only 50% actually got that right. And interestingly, what happens, I will show you the correlation of the iClicker question. And if I put that same question in the exam, it is plus minus 5% of people get that right in the exam. So that is what is called like going into the long-term memory. And I will show you that. So essentially what we are looking at, let's go back to the material a little bit so that we can just recap that. So essentially what you can see that right here, if we're just looking at the transmembrane protein, all of these transmembrane proteins, all of these essentially, except this one, is required, uses SRP, signal recognition particle, and the signal recognition particle actually bring that to the ER cytoplasmic domain. Now, the only thing that is different is that this is a tail-anchored protein, and this one, the gate-3 complex, first it works with a DP, not a GTP, and this is totally synthesized in the cytoplasm, and after that, it is sequestered by the gate-3, and it gets to the gate-3 complex receptor. This is the only one which needs ATP. Rest all 1, 2, 3, except that requires ATP. So very in between lines. You're not going to see this question for the first time in your exam because it becomes difficult, but just remember that. And signal recognition particle receptor is on the membrane. The translocon is on the membrane of the ER. They do not help bringing anything whatsoever. And signal recognition particle works with G, not ATP. So that's your question. All right. So C is the correct answer. So let's go to the next one. All right, this is a little bit bigger, but at the same time, more seconds. All right, good. Stopping it. Good. Yeah, that's the correct answer. And see that how you 74%. So still take a look and make sure that if you just how to answer this question, there's a trick to that. So this question you can answer just by reasoning and remember something from the previous unit. And even if you do not remember all of this. So essentially, if you just look at that, target secretory polypeptide in the cytosolic translating ribosome brings to the translating ribosome to the cytosolic phase of the ER membrane. The next one says targets the secretory polypeptide cytosolic translating ribosome brings to the exoplasmic phase. So these are different. So one of them must be wrong. This, you just do not even take a look at those. Those must be correct because these are two. So that's how you can just answer this question in 30 seconds. So this is one of the things that when you just go to the competitive exam, that you can show, you can eliminate some of the answer choices just by elimination, by reasoning those. So just know that which is the cytoplasmic phase and exoplasmic phase and what exactly is SRP doing. SRP is holding that protein and the ribosome together, and it's bringing in the translogon next to the translogon where you have the SRP receptor. All these receptor and the translogon entrance is on the cytoplasmic side. The protein would go through the translogon into the lumen, which is lined by the exoplasmic phase. So here, all you have to remember that the luminal phase, which is facing the lumen of the ER, that is equivalent to your exoplasmic phase that we learned in Unit 1. So that's essentially for that question. If you just read these two answer choices, you can just make these answer choices. So these are some of the tips that how you should look at the answer and know that what is important to look at and what is not. So that this will help. All right, so let's go to the next one. So this is an in-depth question. So if you have read in between lines, you may get it. This also has something to do with your previous unit, if you remember something. Stop time. Right, stopping it. See, 50%. That is the correct answer. So 32%, 37, and see that this is also another example that how you rationalize the experiments. So let's take a look at this. So synthesis of protein with a high affinity to lipid raft, essentially. So we know from the previous unit, the lipid raft actually has a higher affinity for certain lipids, and those are the clues. You have phenylated protein, acylated protein, GPI. So all these lipid anchored proteins have a higher affinity for the lipid raft's area. Even if you don't remember that, think about that. That SRP, translogon, and the signal peptidase, these actually work with many proteins, including the soluble proteins. Remember that the soluble proteins, which are never going to go to the membrane, that will be brought by the SRP to the SRP receptor. That will be handed over to the translogon. signal peptidase will chop its signal and it will go into the lumen or the cytosol of that. So no protein from a lumen of the endoplasmic reticulum. So no luminal protein which is in the solution cannot be going, will never go onto the membrane. It must be inserted into the membrane before it can go to the membrane. If you understand that, then you will immediately not take any one of these. And even if you don't remember that GPI transaminase or the glycophosphatidylate inositol is a lipid-anchored protein, even if you don't remember that, you can just choose that answer, because these are used by many different types in the soluble protein. So that cannot be the answer. And here glycophosphatidylate inositol transaminase is the answer. So this is how, again, the choice of elimination or reasoning will help you to eliminate these choices. So I'll just go quickly to take a look at that protein, essentially. So, yeah, right here. So remember that this is your GPI transaminase, and if you remember that we already talked about these proteins. So the protein is associated with this, and you have the glucose, mannose, and N-acetylglucosamine with the two fatty acyl chains, and these actually do have very high affinity for the lipid raft. That is not there, but this is something that you learned from the previous chapter. So here you need some connections that what you have learned before. All right, so the next question is based on these topogenic sequences. So we give you a sequence, and you have to corresponding to that, you need to find out that what it is. Let's take a look at that. Yeah, this is good. Okay, all right. Yeah. So this. A couple of seconds. Sure. Right, stopping it. Let's see. Good. Yeah. I'm surprised that this is you guys did your homework well on this one for sure. Yeah. So that's good. So essentially, that how you do this, I will just go over and draw this. That how you will do this essentially. So if you just draw this, essentially, I'm just looking at the NH3, this, then I have a plus, then you have a membrane spanning domain, then I have a plus. So remember that the plus sides will be always on the cytosolic side. So if that is so, I have to draw something like this, and I have plus here. I'm just drawing only two of them. So like plus right here. So if the pluses are on this side, right here, in between them, the part that must be on the cytosolic side. So here, if I'm just drawing it like here, N, H3 right here, and then I have a plus, and so essentially that does not work. So how do we do this? So essentially, if you just draw this, essentially, so you have a membrane spanning domain right here, and then you have another membrane spanning domain right here. So one of them have a plus on each side. So let's do one of them right here, like this. If I just try to do this, I can just draw some plus ends right here, and then I can just draw some plus ends right here. And if I just draw the N terminal on here, N terminal, then going into the membrane spanning domain, and the side of the N terminal is on the side. So this will be wrong. You cannot have that because the N terminal must be facing the other side right here, N terminal. Then you have a plus end right here, plus end right here. Then you have a membrane spanning domain. And then you cannot have a plus on this side. So you have a difficulty doing that. So there is no way. So what you are looking at, that this side should be plus. Essentially, this plus should be plus. So take a look at that how you draw this essentially. So you have an N terminal followed by a plus. Then you have a membrane spanning domain. Here it is plus here, which cannot be true because this side is inside the lumen, which is the lumen side. So this cannot be true. So let's take a look at what happens to the next one. Next one, this is essentially cleaved. So this is a signal peptide, essentially. And that is not in here, that what we're seeing, the N terminal is on the cytosolic side, so it cannot be a signal peptide. Let's see what happens with this one. So if we just draw this one, so you have an N terminal followed by plus, essentially. So N terminal followed by plus, that's good. Then I have a membrane spanning domain. Then I do not have anything in here. That means that this loop is this loop. Then I have another membrane spanning domain followed by a plus on the cytosolic side, and the C terminal is on this end. So that is essentially correct. On the other hand, see that right here, there are things that right here, if we just draw this, the first one does not make sense at all. So that this one, you have a plus side on this side, plus side on this side. Then you have a membrane spanning domain, and you have an N terminal on this side. And then you have this one, and you have a C terminal on this side, which is opposite to this. That is also possible. So the A is not possible, B is not possible, C is not possible because it's the other way. So this is essentially how your sequences are. So just to make sure that, as I said, that all you have to do for this, essentially, sorry. So essentially what you have to do is, let's go to this one. So just make sure that you just get these things actually correct. So that's all you need, essentially. Yep. So that will do it. Sorry. Yeah. Okay. All right. So let's take a look at this essentially. So yeah, so we got this. So good everyone, that how do you do that? Just draw that and just make sure that your plus end you put on the cytosolic side. That's what I wanted to say. Sorry about that. So, I'm just wasting time here. See that the plus are on the cytosolic side, the pluses are on the cytosolic side. See that right here, there is a loop in the cytosol, so you have both the plus are on the cytosolic side. So all you have to remember is that the plus ends are on the cytosolic side. They cannot be on the luminal side, and that will give you the answer. And also remember that is also very important. Think about that. If we just tell that this is a topology of the protein in the endoplasmic reticulum, how this protein will look like if essentially this goes into the on the plasma membrane. Remember that it will look like this. This will be the extracellular place. So this will face outside. This will remain on the cytosolic end, because remember that the luminal space is always equivalent to the extracellular space. That is very important. All right, so that's pretty much it. I mean, like, there's a lot of content in there, and we needed this discussion and the type of question that you can expect. Critical thinking, that's why we spent almost 30 minutes on that. All right, so after that, part 2, there are some of these parts. These are very self-explanatory. That's why I'm not going to talk in detail about that. So you have number of these proteins: binding protein, peptidyl-prolyl isomerase, protein disulfide isomerase. And the first 3, you essentially know that what they essentially do. You do not have to go in between lines. Just know that what they do, just in one word, that stress rotation and essentially rate limiting step. And these are all help in folding of the protein. And then this is a protein disulfide isomerase. You know that there are disulfide bonds in between the chains of the proteins, and they do that. And see that there is a mechanism of that. I did not put any star in there. You do not need the mechanism of this. Just know that what is the function of that? Just using forming disulfide bonds essentially. So that's the only thing. So what essentially this is actually being shown that what it is doing. So this is a protein which is the influenza virus says hemagglutinin. So you know that you always say H1N1, H5N1, and one is H. H is a protein which is a hemagglutinin, which can actually coagulate red blood cells. It can just bind to that. And essentially, when we are not talking about the virology at all here, but when the cell is infected by the influenza virus, it takes over the control of the cellular machinery, and it will use the secretory pathway to make the proteins, and it's an enveloped virus. So it has a membrane on top of it, and this membrane, it steals the membrane from the plasma membrane of the host cell, from where it is budding out. So what it does, it directs the synthesis of this hemagglutinin protein through the secretory pathway, and this is a monomer, and essentially it will assemble as a trimer, 3 copies of the same protein. And this will go from the ER membrane to the Golgi membrane, back to the vesicle, and then transported onto the plasma membrane, where it will assemble onto the virus essentially. So this is your hemagglutinin protein. And what is being shown here, that all these dots that you're seeing, the red, blue, and the orange colored dots that you're seeing, these are the glycosylations. And all these yellowish dots that you're seeing, these are the disulfide bonds. And essentially what is the important part, that what you are seeing, that these are done by this enzyme. Some of these are the peptidyl isomerase, and these are all responsible for these essential. So here we already talked about the binding protein, which actually helps in the yeast, particularly helps in the backsliding of the protein. Remember that. Keep the protein in here. Remember that. And then what we are going to talk about in a lot, so which is the last one, which is the glycosyl transferases. We're going to talk about that a lot. So glycosylation is a huge thing for all the membrane proteins. So here you can see the function of the PEEP, which binds to that, prevents backsliding if it by chance happens. We are going to talk about these dolichol oligosaccharides a little bit later. PDI is forming the disulfide bonds, and we're going to see that this is a calreticulin, and this is a calnexin. These two are the quality control mechanism for the proteins. They check if the protein is folded properly or not, because they are spatially arranged in such a way that if they cannot bind into certain region of the protein, they can mark the protein as not properly folded. It will go through the cycle again, and if ultimately it cannot be folded, we're going to see that there will be an unfolded protein response to degrade that protein. And ultimately, this is just only one monomer of it. So you can see that this is essentially the membrane spanning domain. This is a huge N-terminal in here, and this entire part will stick out of the membrane. And when 3 of them assemble, this is an HA0 trimer. And again, it cleaves a disulfide bond to form a fusion peptide and becomes H1, not needed. But this will stick out of the influenza virus, essentially, and this is the one which binds to the receptor. So that's basically the synthesis of that. Detail is not necessary, but just showing you that, say, BIP, PDI, and then glycosylation that we are going to see, that these are all essential for the membrane proteins. Almost all is 99.9%. I do not know that whether there is an exception or not, but all protein which are outside the cellular membrane are glycosylated. There, probably, I mean, like I cannot just think about that one which is not glycosylated, because glycosylation plays a huge important role, and that's why we are going to see these glycosyl transferases after this. So what happens that this is, you see that all these proteins actually work to fold the protein once it actually comes into the ER. It could be either in the solution, which is a soluble protein, or it's a membrane-bound protein. But if the protein cannot fold properly, what happens? So know this very well, because we will have a quiz question out of that. So there is a quality control machinery in here, and also a degradation machinery. So on the ER. So what you have here, these are unfolded proteins, and these are soluble proteins. Just, it could be membrane proteins as well. And there are some endonucleases here, which are called IRE1 monomers. And these IRE monomers means like a single protein. It is associated with the BIP, or the binding protein. And when they are bound with the BIP binding protein, they do not interact. And this IRE, although it's an endonuclease, that means that it cuts certain part of the nucleic acids that is present in the cytoplasm in certain sequences. So when they are monomer, they are inactive. They cannot cut or digest any nucleic acid. But what happens that if there is an attempt that the protein tried to fold and it did not fold properly, then these binding proteins bind to these proteins. This is called an unfolded proteins with the BIP bound. And this caused what we call as an unfolded protein response. So these BIPs, which were actually keeping the IRE monomers separate, the BIP comes out, and now they can interact. Now remember that this is entirely different from the BIP, which does have a different function of preventing the backsliding. That's a different story. This is a different story as well. So two are entirely different part. This is BIP is helping to dimerize the IRE to make it active, so the the endonuclease activity actually works. So now this part, a lot of people actually confuse that, and let's see that where this is where your basic idea of your DNA replication, transcription, translation comes in. So in the cytoplasm, you have this HAC1 mRNA. Now, if you remember from just a little bit basic knowledge of your transcription, in eukaryotes, the mRNAs do not come out as mRNAs are not produced as mature mRNAs. What I mean by this is that in between them, you have some region like this, which we call as an introns. Remember that these are the introns. I'm just writing introns. Whereas the red ones, that what you are seeing, these regions are called the exons. I'm just writing exons. So the exons, you have 1, 2, 3 exons, and you have 2 introns. The introns you can think about, although we know that there are now some implications of that, are essentially junk sequences. They do not code for anything. The exons actually code for the protein. So in the nucleus, you produce the mRNA like this. And then this part essentially is spliced out. And your exons 1, 2, and 3, 1, 2, and 3, which were the red part 1, 2, and 3 is joined. And this is your mature mRNA. And this is transported into the cytoplasm. That would be read by the ribosome to make the protein. mRNA, there is an exception. The HAC1 mRNA is not fully spliced. It does connect, and right here is the intron, which does have a junk sequences. So this mRNA cannot be translated into the protein, but it is produced like that. And one of the exceptional mRNA that is transported, most of the mRNAs unspliced will not get transported outside the nucleus once it is produced by the RNA polymerase. It will stay there. But the unspliced mRNA, this is an exception, that will come into the cytoplasm. But it cannot do anything because this unspliced mRNA, ribosome will not read that, make proteins. Now, when you have a response of this unfolded protein response right here, IRE1, this endonuclease will essentially cleave this part, and then this will be joined essentially. So this is the splicing effect of the IRE1. So now you have the HAC1 mRNA. This can be translated by the ribosome, and this will produce what is called a HAC1 transcription factor. And then a lot of people say that this is making a transcription factor, and this is out of the ER and degraded. But remember that this is just a transcription factor. There is a whole lot of thing going on after that, which is not in there. So just make sure that you get that. So right here, I'm just drawing the nucleus. So this is the nucleus of the cell. So this is just the part of the story. This HAC1 transcription factor is a DNA binding protein. Remember that it is, you can write down that this is a DNA binding protein. And this is just a factor. This will go to the nucleus, and you have the DNA. It will bind to several regions of the DNA. That will transcribe mRNAs, lots of different mRNAs, which code for the degradation machinery. These mRNAs will come out into the cytoplasm. All of them will going to make separate proteins. All of them are going to make separate proteins, and those separate proteins are essentially, say, proteasome, ubiquitin ligase, many different ones that you can read your textbook. They are in detail. And those proteins will essentially degrade the unfolded proteins out of the ER. So just make sure that you just only get that the HAC1 transcription factor is just the tip of the iceberg. It is essentially the signal to make a number of DNA sequences transcribed into other mRNAs that will be translated into the degradation machinery proteins. So that's the entire story. So just make sure that you spend a good amount of time so knowing this slide and know the steps for this for sure. All right, so let's take a look at this. We just got this one, so this is fresh out of your mind and you should be fine. Not out of your mind, in your mind. Right. All right. Good, everyone. Last chance. Stopping it. Yeah. that is correct as well. So just remember that what is happening, dissociation of the binding protein from IRE monomers, subsequent dissociation of the folding proteins, that is the first thing happening. So look at where is 4. So C and A is possibility. So let's take a look at what is happening after that. Then dimerization of IRE, that is correct. So these are going well. Now let's take a look at this, the translation of hack mRNA into the transcription factor, then splicing of the unspliced mRNA. Before the translation, the splicing must happen. So it's 3, and then translation of that happens. So very straightforward. So just know, as I said, know the steps, and you should be good. All right, good. Let's go into the glycosyl transferases. Now, these glycosyl transferases, this is a generic name. There are hundreds of them, and they do all different things, and we are going to just touch the tip of the iceberg with some of them. So, one of the most common type of glycosylation that we essentially see is what we call as an N-linked glycosylation. Now, here, tell me that anyone, this is a wording, what do you mean by an N-linked glycosylation of a protein? Where in the protein it is glycosylated? Anyone? What do you mean by an N-linked glycosylation? Yeah. What? Yeah. That is a correct. Make sure that sometimes I just assume that all knows, but sometimes I find some of the question that is asked. Remember that N does not mean the N terminal of the protein. N is a single letter amino acid representation of asparagine ASM. So just make sure that you get that one. Good, very good. Thank you. And so essentially anywhere in that, so this NH2, this is your N terminal. And the glycosylation is not happening in there. It can happen there as such, but it is not happening, and we do not consider this N-terminal glycosylation at all, or C-terminal glycosylation. There's only 2 types, the N-linked and O-linked. We are going to see that later. So this can happen anywhere, but you have to have a kind of a signature sequence where you have an asparagine, then X means any protein, and then serine and serine or threonine. If you have that sequence, that is essentially a glycosylation site. And this is essentially that you are looking at the asparagine side chain, which is N single letter, and this is a glycosylation pattern. So you can see that there is a typical type of glycosylation where it happens in acetylglucosamine, and then 2 of them. And then you have 1, 2, 3, 4, 5, 6, 7, 8, 9 mannose, and then you have a 3 glucose. And we are going to see that when the glycosylation happens, there is a lot of extra amount of these sugars. All of these are sugars, remember that, carbohydrates. They are put on, and then gradually they are trimmed. And that process essentially enhances the folding and quality control. That's what we're going to see. So let's take a look at how this essentially happened. So there is, like, you do not have to memorize this entire scheme. Just remember that. So essentially what you are looking at in here. So here, there is a lipid right here. It's a phospholipid. This is dolicol phosphate. Definitely remember that. Plays a key role in glycan precursor synthesis in glycosylation. This lipid is essentially the base on which a glycosylation precursor is prepared. And very interestingly, we are going to see that. So what is happening in here, I'm just going one at a time. This is phosphorylated. So here an UDP actually phosphorylates and put like an UDP residue in here. And now you have 2 of these in acetylglucosamine. So right here is use the UDP, that the energy from that, and you put 2 phosphates followed by 2 in acetylglucosamine on top of that. And then you put gradually, with the help of GDP, a number of mannose. So here you have the 2 N-acetylglucosamine, you have 1, 2, 3, 4, 5. When there is a 2 N-acetylglucosamine and 5 mannose stage, this dolicol phosphate is flipped inside. So what is dolicol phosphate? What kind of lipid? Is going from the outer membrane to the inner membrane, taking the precursor with it. Anyone? We talked about that if a lipid actually comes from this one end to the other, no? It's a flipase. So the glycol phosphate is a flipase, and the first step, which is the addition of the N-acetylglucosamine, is blocked by tunicamycin. So if you're just doing your lab, you're going to see that you use this antibiotic, which will essentially block any kind of glycosylation in the ER. So this is basically cause the ER stress. No glycosylation is going to happen. So make sure that if you just remember that this is the molecular function of tunicamycin, the very first step of blocking glycol phosphate. So now that 5 nano stage is brought inside. This is the cytosolic surface. So on the other hand, the glycol phosphate, see that what it's doing. So I'm going from here to up to this, and then I'm starting in here. It brings another glucose with the help of an EUDP, and just one glucose, it actually is flipped inside right here. And see that on the other hand, there are others which are bringing in more mannose, single mannose. So this mannose is bound and it's bringing another mannose. That mannose is put in there, and gradually this is happening number of types, and then gradually this is increasing from 5 to ultimately 9 mannose. 1, 2, 3, 4, 5, 6, 7, 8, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 mannose right here. Am I counting correctly? Or 9? 9, right? Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah, it should be 9. There is never 10. So, yeah. So 9. So 5 to 9 mannose. Now, on top of that 9 mannose, these glucose which are brought in, those are put on one at a time, and it's a 3 glucose at this step. So essentially, how the uridine is bringing these things, essentially, you can see that it is a nucleotide. This is uridine, this is the ribose sugar, and to the phosphate a sugar is associated, and that is essentially brought in if it's an acetylglucosamine or the glucose. Only the mannose is brought with the help of a GDP, where this will be a guanidine, essentially right here, and this will be the mannose. So this is the overall idea. So this is the entire precursor. So you have 2N-acetylglucosamine, and of course this is phosphorylated, the lipid, 2N-acetylglucosamine, 9 mannose, and the 3 glucose. And this entire part from the glycol phosphate is going to be transferred to the asparagine. That's what you are seeing. So this is the membrane protein, glycol phosphate associated with this, and glycol phosphate will be again recycled back. Now, what we are going to see, that you have 2N-acetylglucosamine, 9 mannose, 3 glucose. They are going to be trimmed by these different glycosidases. So they are going to digest these, and it's going to have a different form, and that is essential for the folding and quality control of the protein, and we are going to see that. So what you are going to see, that right here you have the 2, 9, 3. That is essentially the structure. And then gradually you are seeing that it is essentially trimmed, and all the glucoses are gone. And here you have 2N-acetylglucosamine, and you have 1, 2, 3, 4, 5, 6, 7, 8 instead of 9. Once this trimming reaches 2N-acetylglucosamine with 8 mannose, this is ready to go to the Golgi. If this protein is a protein which is a resident protein of the ER, then it will remain in the ER, or it's a resident protein of the ER membrane, it will retain in the ER. But if this is a soluble protein or membrane-bound protein, and if it is supposed to go to another destination, this is where it will go, because this will be essentially packed into the transportation vesicles, essentially. So from here, it is trimmed one at a time. So remember that essentially. So where are the destination? So here what you are looking at, that this is the endoplasmic reticulum, and this is essentially the transphase from where the vesicles are being formed. These vesicles are going to go to the Golgi and fuse at the cis-Golgi network, and then from each one of these stacks of Golgi, it's going to form a vesicle, go to the next one, go to form a vesicle, go to the next one, and go to ultimately the trans-Golgi network. In each of these stacks, there are going to be modification of further modification of those glycosylations. And then once that glycosylation is done, and that will be a signal essentially from the trans phase of Golgi, it does have three destinations. It can go to a lysosome, essentially. It can go into the plasma membrane. It can carry the plasma membrane protein in this in the membrane of these vesicles, which will essentially fuse, or it can have soluble proteins which can be secreted outside the cell. So these are the three destinations from the Golgi. But in between the Golgi stack, there are different enzymes in each stack that's going to modify the protein in different ways. So this is essentially the way it works. So this is very straightforward. So let's take a look at this. So this is the cis phase, that is entry, the trans phase, that is an exit. So we saw this picture sideways right here, and this is up and down. Remember that cis phase entry is from where the vesicles from the endoplasmic reticulum come in. So this is exactly what you are seeing right here. Now, what we're going to see, essentially, we're going to, I will come back to the previous slide, just in a continuation, remember that. Right here, this part is the ER. Now see that these are the different Golgi stacks, and this is the trans-Golgi. And each step does have different enzymes and a different modification of this glycosylation. And this is a huge part of this part 2. So just remember this part 2 very, very well. So let's take a look at this, what is happening right here. So because this part is needs some good explanation. So here, what we are looking at in this part is that this is an end-linked oligosaccharide processed into two types. One is called a complex, and one is called a high mannose. Both of them are there associated with asparagine N. So here, if you have a complex, see that what is the structure? You have 2N-acetyl-glucosamine, 3 mannose, then 3N-acetyl-glucosamine, then you have 3 galactose, and then you have the red ones are N-acetyl-raminic acid, commonly as sialic acid. This glycosylation is a complex glycosylation, whereas some of them could be just N-acetyl-glucosamine and number of mannoses we call the high mannose. So where you get this? These modifications are going to happen step by step in the different Golgi compartments, and the matured protein. Matured protein means that this is actually coming out from the trans phase of the Golgi. When the proteins are completely matured in the trans phase of the Golgi, you are going to see these type of glycosylations if they're end-linked. Make sure that you understand that. So this is nothing to do with the ER. We actually stopped at the ER, that right here. This was what was going into the Golgi. Just only 2N-acetyl-glucosamine and 8 mannose. And in Golgi, all these things are going to be trimmed and taken off, and they will be added with these or these, essentially a high mannose and N-acetyl-glucosamine. And this is what we call as a complex glycosylation. And this can go into the plasma membrane. This can go into the lysosomal membrane, and these can be secreted as a soluble protein as well. So these are the different destinations. So this is again another rendition, that processing of the end-linked glycosylation. So you can see that this is the same picture that we have before. You have the 2, 8, and 3, and then you have just 2, 9, and 3, and then you have 8. It's going to go to the Golgi. And why this is important? Because these are the calnexin and the calreticulin. All the way back that when we saw the hemagglutinin, the HA folding, you saw the name of those two, CNX and CRT, calnexin, calreticulin. These are quality control proteins. They actually bind to these glycosylation and do the quality control. And if you see that they have not properly trimmed, they will not be properly folded, and they will be trying to properly trim those back again. If they are unable to, then what happens? It decreases degrades it, and then it just goes into a degradation path. So essentially, all you have to take home a message from here is that calnexin, calreticulate, these are essentially quality control protein to check proper folding and glycosylation of the BR proteins. If they cannot achieve that, it will be degraded by a protein complex called OS9. Number of proteins, what is not needed. So this is the same thing that is happening here. That is a proper glycosylation. And then now we are coming into the Golgi. So remember that we came to Golgi with 8 of these mannose. Now if we just go to the Golgi and start with the Golgi, see that the first step, the Golgi mannose 1 removes 3 mannose. So from 8 it becomes 5. And see that now it is putting 1 N-acetylglucosamine on top of the mannose right here. And at this point, it crosses approximately the medial part of the Golgi. And after that, you are going to put these subsequently before it reaches the transphase of the Golgi. So this is a moment that make sure that you get this very, very clearly, because this is where people get confused. See that what is adding in here. After this, and see that this picture is, the asparagine is at the top, and the glycosylation is on the facing towards the bottom. The previous picture that we have actually seen of this, asparagine is, this essentially is on the sideways. Remember that. The asparagine is like this, and the glycosylation is sideways. And take a look at that. 2 N-acetylglucosamine, 3 mannose, and after medial Golgi, you have put 1 N-acetylglucosamine. You are going to put 2 more glucosamine, 3 galactose, and 3 sialic acid. So where that corresponds to it here, right here. So add on top of that 1 and 2 N-acetylglucosamine. On top of that 3 galactose and 3 sialic acid. So make sure you connect this thing to the matured protein that is going to come out of the Golgi as a complex. That, just make sure that this 2 connection is very important. So that is what you are going to get in the Golgi. Now, this is what it is. See that right here, oligosaccharides with the lysosomal, this is lysosomal proteins, removal of the mannose, addition of N-acetylglucosamine, addition of galactose, addition of the N-acetylgluraminic acid or the sialic acid. So this is essentially telling you where the complex proteins are being essentially formed, and connect that with those slides beforehand. So this is the, now what the important part is that this can be tracked. If it is a complex protein right here, complex protein, we can track that when it is passing the medial Golgi. The reason is that there is a glycosidase, which is called endo-H glycosidase, and this is actually used in the protein trafficking study. Before the addition of this N-acetylglucosamine, just digest this protein, is going to chop the glycosylation from here. The protein remains, that glycosylation goes away. As soon as it just becomes a complex protein right here, it is resistant to endo-H. This cannot be cleaved by the endo-H. So if we just do a time point of the trafficking study, that when they are passing through, you can actually essentially tell when it passed the medial Golgi for the trafficking studies. This is one. Another example is that this is an entirely different protein, like glycosidase, endo-D. This can tell you if your protein, different question. The previous question was when it is passing the medial Golgi. Here it is addressing a different question. Here it is telling you when it is in the ER and when it came to the Golgi. Because see that what happens in the ER, you have 8 mannose, and this cannot be cleaved by the endoglycosidase D. When it comes to Golgi, it becomes trimmed to the 5 mannose, and see that right here, 3 mannose is gone, it is 5 mannose, so that means that this is the first Golgi enzyme and it's 5 mannose. And this glycosylation can be cleaved and cleaved from the protein. So what you are seeing in this process? So these were a trafficking study that was done in a glycoprotein of FS stomatitis virus as it is moving through. So you, essentially what you do that you label the protein and with a 35-S-methionine or cysteine, and then you take the sample every 5-10 minutes, immediately plunge it in the ice and then isolate that organelle and just look at those, and then you do an precipitation reaction. Usually you should have an antibody against that by which you can pull them. So what you are essentially seeing at the 0 minute, see that there is a band, and the thing is that from here it is probably not very clear. You're thinking that the band is getting thicker. No, there is a doublet. There is a separate band, and the bottom band is actually getting thicker and thicker. So what does that mean? That at the initial stage, the molecular weight is slightly higher because this is the molecular weight of the protein plus the glycosylation. And as time goes by, the proteins are transported from the ER to the Golgi. In Golgi, the endoglycosidase is removing this residue or the glycosylation. So it's only protein, and the molecular weight of the protein is less than with the glycosylation. So gradually, the bottom one is getting fatter, and the top band is getting thinner because more and more Golgi is being transported from the ER to the Golgi, and this is essentially what it is. So this tells you endoglycosidase whether it is in ER or the Golgi. Endo-H tells you where you have that essentially the protein in your in the ER. So here we are looking at, and remember there are two slides, and we'll spend some time with that. You can have as much as 10 questions out of that. So basically, big question, 2-3 questions, like 4 questions probably just related to that by interpretation of data. So just make sure that as much as 10 points. So make sure that you understand that. So here we are using the endoglycosidase H in the trafficking studies to assess the movement of glycoprotein to the ER and the Golgi. Before we go into this slide, we'll go to the next slide to have some basic information. This is another very ideal word, that the endoglycosidase H can distinguish between the high mannose and the complex sugars. What it means. So we have the 2 gels, and here I have taken the protein. Remember that whenever we are doing these studies, let's go back to the ER. Whenever we are doing these studies, we are taking the protein, all the way matured proteins from here. And what are the 2 different proteins? Remember that these 2 proteins can be either complex or high mannose. They are completely done, glycosylation completely in the ER, in the Golgi. So we are taking those proteins, and one of them I'm not adding any endo-H, the enzyme, and another one I have added. Here, no endo-H added. So if you look at that, then the completely formed high mannose, the residue or the glycosylation is digestible by endo-H. So the endo-H is always digestible by this, and as the glycosylation is removed, this protein is with the glycosylation, this is without the glycosylation. But see in the complex. The complex protein cannot be digested by the endo-H, so this is with glycosylation. There is no lower band in there. So if you take a mature protein out of your Golgi or in your plasma membrane and digest it with endo-H, just looking at this pattern, you can tell whether your protein is a complex glycosylated proteins or a high mannose protein. Everybody good with that? So if you see a lower band, you can tell that is your high mannose. If you do not see both the proteins are at the same level, you could not remove the glycosylation, so they are at the same level. On the other hand, now we can do a trafficking study. Second set of experiment. First set, distinguish between high mannose and complex. Here, we are looking at the studies in every 15, 30, 45, 60 minutes. And as it passes through the Golgi, we wanted to know that when this complex glycosylation actually happened. So if you just go back to the picture right here, see that the glycosylation happens just past the medial Golgi right here. So we are going to see that time point from this experiment. So you can see that right here, this is digestible, this is digestible. So the modifications that you have is digestible at this point. Here, they become resistant, the glycosylation become resistant. So we can tell that in between about 30 to 45 minutes, the protein actually passed the medial Golgi. So now what will happen, how this picture will look like. So this is your complex protein with the complex sugar modified proteins. And now tell me that how this will look like if we take a high mannose protein, high mannose glycosylation proteins. What we should look like. Anyone? Instead of complex, you can see that right here, it becomes resistant in there. And how would it will look like if it's an H? Anyone? Okay, all right. So let's go to this one. We already gave you that idea that this is a high mannose, completely matured, complex, completely matured. So what you will going to see that in here, it will be always digestible. If it is high mannose, you cannot tell when it is crossed the medial Golgi. So remember that endo-H, if you are using endo-H, you can distinguish which is a high mannose and complex. But you can do the trafficking study only with the complex protein. You cannot tell it with the endo-H because it will look like this the same way. So if you have a panel like this without these pictures, and we ask you that tell me that when it passed the medial Golgi, suppose A panel, I give you this experiment. This is your A protein, this is your B protein. And then for the A and the B protein, I give you this panel. The B protein looks like this, and the A protein looks like this, and asking you when it passed the medial Golgi. For the A protein, which is high mannose, you cannot tell. Remember that you cannot tell the trafficking. You can only tell it for the complex. So we spend a good amount of time on this one because this is where most of the people actually do struggle. And so that's one of the important points in here. All right, so these are some of the secretory proteins that from ER to that you can just track with that. And the last thing that what we are going to see from here, that there are some proteins which are resident proteins of the ER. And these proteins do have a sequence which are called KDEL, essentially. So it's lysine, aspartic acid, glutamine, and leucine, KDEL. And these proteins, sometimes they are supposed to work in the ER, but they are sometimes packaged into the vesicles, and they go into the Golgi. And that is a loss. This is totally by accident that this happens. But there is a mechanism by which they can be brought back into the ER. So what happens that in the endoplasmic reticulums, the pH is slightly alkaline, about 7.2. Here it drops to about 6.8, slightly acidic. So in here, there is also a KDEL receptors. These KDEL receptors, along with the KDEL proteins, are leaked out into the ER, from the ER to the Golgi. In the alkaline compartment of the ER, the KDEL protein and the KDEL receptor doesn't have any affinity. But in the acidic pH, they actually have essentially affinity. So the KDEL receptor in the acidic pH capture the KDEL proteins, and once this happens, it triggers a vesicle formation with the COP1 coated vesicles, what we said, and they are going to return back into the ER. When they go forward, they are coated by a protein which is called COP2. When they are returning, they are coated by a COP1 vesicle. And we'll stop there and we will talk about the trafficking. So make sure that you learn these topologies very, very well. We'll take it slow and then we'll pick it up.